Okay, so first we started with the innate system. Almost steady. Okay, so remember your first line of defense is skin, mucus, normal flora, and those are all barriers. And then both of these are second lines of defense. And that's going to be saliva, tears, pepsin, sweat. We added in fever. We added in inflammation. Right here, so inflammation. Okay, and then your second line of defense is your phagocytes. So really what that is like, if a virus were to get through number one, get through the saliva, get through the fever, then it's up to the phagocytes to take over. Okay, and then the phagocytes, we know, have the MHC2 molecule on the surface with its antigen. What does that do? What's the MHC2 do? Good. It holds out the antigen to show other white blood cells, right? To show other helper T cells and other macrophages. Okay, and then it also, the phagocyte releases the IL-2. And what is IL-1. What is IL-1?
It's like the same as macrophages.
Good. Is it?
Wait,
Yes, chemical. Say it again.
IL-1, what does it do?
It's like the chemical that triggers the helper T cell.
Perfect. It's the chemical message that activates the helper T cell to tell it needs help. So the IL-1 activates helper T cell. Helper T cell releases IL-2. And IL-2 goes to the killer T cell, which is right here. And to the B cell, which is right here. Okay, so, and I said that all this is reception, it receives the message. Transduction, it prepares to release IL-2. So the response of IL-1 is to release IL-2. Does that make sense? Okay, so IL-2 is released from the killer T cells, and the response, so reception right here, reception, transduction happens on the inside, and it releases this toxin, paraforin, and that penetrates the cell membrane of other cells, of infected cells. So killer T cells, that's cell mediated. It kills infected cells. Okay. Humoral is in the blood. So the B cells release antibodies. You can see my antibody right here. B cells release antibodies. And antibodies do what? Antibodies do what? Come on. Attached to the antigen, where at they? On the pathogen. Good. So T cells right here kill infected cells, while B cells attach to pathogens so that they're easier to identify. Does that make sense? So all this is cell-to-cell communication. This is your third line of defense. If it gets through the skin and it gets through the macrophages, then it's up to the T cells and B cells to handle it all. Okay, this picture, this diagram right here, I designed. Like when I didn't understand the immune system, when I first started teaching this, I drew this out to help me understand it. And so that's how I taught it. But your textbook author thinks that this is the way to draw it. So this is how you're going to see it on the test or the AP exam if you ever see it. So take a second and look at this and see if you can make sense of this. So y'all take a couple minutes and do that. Okay, so the first thing it says is, or the first thing I see is the first exposure. Okay, so when the first exposure, that's like the first time the organism comes to you, it's up to the macrophages to start. So if I were you on yours, I would draw a little MHC molecule right there. Dadgummit. Draw on your paper you have, draw a little MHC molecule, just like we did. There's your little MHC2 molecule. Okay, so remember that macrophages present the MHC2s. So we're just going to add a little detail to this picture. They present the MHC2s so that helper T cells can see it, and so that other macrophages can see what they've killed, identify what they've killed, and they can go kill it too. Okay, then macrophages signal to helper T cells. What is that message called? When macrophages signal to helper T cells? Interleukin 1. So write that in on your paper. Write in right here, interleukin 1. Okay, so macrophages release interleukin 1, which tells helper T cells to release what? What do helper T cells release? Interleukin 2. So write in interleukin 2 on both sides. Okay, so interleukin 2 is released from the helper T cell, and it goes, let's go B cells first. So it goes over here to the B cells, and these B cells your textbook calls naive B cells. It's like the B cell that doesn't know how to make the antibody yet. It's the B cell that's still out feeling, wondering how to make it. It's out using its glycoproteins, feeling. So the B cell eventually forms, and this shows, it says gives rise to a working B cell. And a working B cell is one that's making antibodies, and it calls that a plasma cell. But I've also heard that called effector cell. So you might want to write over to the side an effector cell. So a plasma cell, an effector cell, those are just B cells that are actually working, making antibodies. And you can actually see the antibodies down here. Okay, and somebody tell me what antibodies do. Attached to the pathogens. Okay, read this bottom part right here. This is important. It's just written so small, I don't know why. Read that bottom part down there. It says B cells. It's talking about the B cells. They defend against extracellular pathogens by binding to an antigen. Somebody tell me, what's an extracellular pathogen? What would it's a pathogen not inside the cell. So that's all it's saying. B cells bind to pathogens not inside the cell. They're extracellular. And it says, and making the pathogen easier target for the phagocyte. So all it does is the antibody attaches to the pathogen so that it's easier to identify for the phagocyte. So they just wrote it a little differently. Okay, now let's go over to the killer T cell side. So they called it a cytotoxic T cell, killer T cell, same thing, that's activated by what? What activates that killer T cell? Interleukin 2, good. And that is going to make, it calls it an active cytotoxic T cell. Again, that's an effector T cell. It makes a T cell that's working. And what does that make? It doesn't show it, but what does that make? Yeah. Good. Okay, and then that they both give rise to memory cells. So, and remember, memory cells aren't necessarily making antibodies or making paraffin. Memory cells just know how to make it, so they're just swarming the body looking for that. Okay, now read this bottom part where underneath the T cells, it says, defend against intracellular pathogens. What's an intracellular pathogen? A pathogen that's already infected a cell. So it defends against infected cells and cancer by binding and lysing. What does the word lysing mean? Breaking apart, right? By binding to it and blowing it up. Remember, like with the big guns from that video. Okay, remember the difference in cell-mediated and humoral. Somebody tell me what's the difference. Good. This is cells over here. This is the humoral is blood. The pathogen in the blood or the pathogen in the cell. You think you got it? Okay, good. Okay, so here's the math quiz. Okay, notes. Go ahead and pull out this last part for the immunity. Okay, so the first part of the notes, talks about long distance, paracrine, which is local, and direct contact. So if you look at this picture right here, it shows you this first one is in direct contact. And remember us talking about how plants did direct contact? What was it called when plants communicated through those vessels? Plasmodesmata. Remember that? Maybe? Okay, so this is just showing you direct contact. When the glycoprotein touches the MHC1 and 2, that's direct contact, cell-to-cell direct contact. Okay, when paracrine would be local contact, and that was like local signaling, and our example when we looked at that was actually neuron, so that was local. And then long distance is when it goes in the blood. So I think IL-1 and 2 would be long distance, when that's going out into the bloodstream. Okay, the next part of the notes talks about self-tolerance and AB blood types. So look at this picture with me. We're actually going to fill it out together. Okay, so somebody with A-type blood has a cell membrane on their red blood cells that have A antigen. What is an antigen? Somebody know what that is? It's a protein that would actually make antibodies come. But it's a surface protein. Do y'all remember seeing that? Okay, so an antigen is just a surface protein. Well, people with A blood have proteins that are A proteins. Okay, so people with B blood have proteins that are, I'm going to draw triangles or try to, that are B proteins. So it's just a different kind of protein. It's not that one is better than the other. A blood and B blood, it's just they have different surface proteins on their red blood cells. Okay, people with AB blood have both A and B. Can y'all see this? Does this make sense to y'all? I should have made different colors. So they have the circles and the triangles. So AB, it's not that any one is better than the other. It's just a different protein. People with AB have both of the proteins. They have both A and B. Okay, people with O blood don't have any surface proteins. And that's not a bad thing. They just don't have any surface proteins on their red blood cells. Do what? Right, so A has a special protein that's different called an A protein. B has a special protein that's different called B protein. A, B have both A and B, and then O doesn't have either antigen. Okay, so people with A blood have the B antibody. What is an antibody? It attaches to the antigen, right? So people with A blood have an antibody floating around that's for B protein. So what would happen if they got a blood transfusion that had B in it? Their immune system would attack, right? So if you got the wrong blood type, what would happen is your blood would start to clot and your immune system would start to attack and you get really sick. You go into shock, actually. Okay, B blood, they have A antibody. I'm going to draw an antibody this time. People with AB blood have no antibodies. They have no blood antibodies. So they don't have an antibody for A or B. Why? Yeah, because they have both A and B. Okay, we're going to talk about that too. Okay, people with O blood, they don't have any antigens. So they have both antibodies. They have A and B antibodies. Mm-hmm.
So the best one to have is AB.
Well, it depends on, you say the best one to have, it depends on if you need blood or if you're given blood. If you need blood, the best one to have would be AB. Okay, so let's go with, A can give blood to who? A can give blood to A and AB, right? Because think about it. If you have this kind of blood right here, look at it. You have triangles and circles. And you get this blood inserted, then the glycoproteins are going to feel those circles and feel that it's normal and be like, oh, nothing unusual about that, right? Because your body fights foreign pathogens. Your body is going to attack different antigens. But if it's the same antigen that you've got, then it's not going to infect it. Okay, so people with A blood can receive blood from A and O. They can get A and they can get O. Okay, if you have B blood, you can receive from B and O. Right? Can y'all see this? Okay, people with AB blood, who can they receive from? They can receive from A, B. They can definitely receive from AB, and they can receive from O. So people with AB blood can receive blood from A, B, and O. So we call that the universal receiver. And then O blood can give to everybody, but can only receive. So O blood is like someone who is really, really, really generous. They give, but they can't receive. At Christmas time, you have to give presents, but you don't get any presents. Okay, AB is opposite. AB can receive presents, but they never can give. So I have O blood. So can I give or receive? I can give, but I can't receive from anybody but O. Does that make sense? Okay, so they figured all this out, and they were like, "Oh wow, well this is perfect." They figured out how they can do blood transfusions, but about 30% of the time it would fail, and the blood transfusion wouldn't work, the blood wouldn't take, and the person would go into shock. So they were studying the rhesus monkey, and they figured out there's another protein. It's not just A and B. There's another protein, and it's actually called the RH factor. R is a big R and a little H factor. And so instead of saying you have ARH or just regular A, they say A positive or A negative. So if you've got the RH protein, it's positive. If you don't have the RH protein, it's negative. Does that make sense? Okay, so I am O negative. That means I have no surface antigens, and I do not have the RH protein. If I was O positive, I would just have the RH protein, right? Okay, does this make sense? Yes? Okay, so now we know how to do blood transfusions just perfectly. Okay, so watch this, you want to draw this out probably. If a mom is pregnant, this is a pregnant mom right here. Yep, she's a pregnant mom. And she is negative. It's important to know that the mom is negative. Okay, it's important to know that the mama is negative. So if you're drawing this out, either right above it or draw a negative sign on her forehead like I did. That is not a unibrow. That's a negative sign. Okay, so the mom is negative, and she has a baby. Let's say she has a baby that has a positive blood. The mama and the baby don't have to have the same blood. Blood is just like any other gene. It's inherited from both the mom and the dad. Okay, P.S. Ready? Somebody with A blood either has 2 genes for A, or they have a gene for A and a gene for O. That's both A blood, because O is recessive. Right, so B blood is the same way. B, or BO. Somebody with AB has to have an A and a B, because A and B are both dominant. This is where there's more than one dominant allele. Remember alleles from 9th grade? The letters? Okay, so someone with O blood has to have both recessive, and you just do a Punnett square. So it's possible for the mom to be negative and the baby to be positive. Okay, back to my mommy story. Y'all got a fat mommy drawn over here? Okay, mommy is negative, has a baby that's positive. No big deal, everything's perfect. Baby's born, the little positive baby, that's the first child, is born, and everything's great. Except when the baby was born, during the childbirth, a little bit, just a little bit of the baby's blood got into the mom. Now the baby has skin, so the baby doesn't get any of the mom's blood. But a little bit of the baby's blood got into the mom. So a little bit of a foreign antigen, the Rh positive, got into the mom. The baby was born. So what did the mom's immune system do? Attacked it. It attacked just that little bit of blood. It got rid of it. It was fine. But the mom formed memory cells. Memory B cells, memory T cells to kill that blood. It attacked it like it was a virus or something like that. Okay.
So she got pregnant.
So she got pregnant again. When,
Like eat the baby?
when baby number 2 comes around, if baby number 2 is positive, assume it's the same dad. That's a nice assumption. Okay, assume that baby number 2 is also positive. Baby number 2 has extra arms or something. Okay, assume that baby number 2 is also positive. When baby number 2 starts to develop the blood, the mom's body, those memory T cells and B cells, would attack the baby and kill the baby. So, how can we fix that problem?
You can't change the baby.
So does that not
You can't change the baby's blood type. It doesn't happen because we figured out how to fix it.
You can like put her in the background.
Okay? How could you trick those? How could you trick them? Lex is on it. She said you could trick the white blood cells, but how could you trick them? The problem is that those memory cells are in the mom's body. So you've got to prevent those memory cells from ever forming. Do what? What'd you say? Yeah, no, you can't fix the antigen on the baby's blood. But what they do is when the baby number 1 is born, they give mom a shot that suppresses her immune system, that weakens her immune system. And so you have to give the shot within 24 hours of childbirth. So if the mom is negative, they give the shot within 24 hours of childbirth, and that's going to suppress the mother's immune system so she doesn't form those memory T cells and B cells. Yep, and the shot is called Rogam. Have you ever heard of that?
Has everyone got it or just... Wait, is that the little thing that get the little spot on the...
No, that's like that's the polio vaccine, I think. But it's just a shot that they give. You probably get lots of shots after you have a baby. I don't know, I've never had one. But I feel like you would get lots of shots. That's just one of the shots you have to get is a Rogam shot. And I know they give it to moms who are negative. But I don't think they give it to everyone. They give it to moms who have negative blood type. So when baby number 2 comes around, it won't be a problem. But they have to do it every time the negative moms have a baby.
I thought it was oh, never mind. So if the mom's positive,
If the mom's positive, nothing happens. The mom's positive and the baby's and negative, nothing happens. Because the mom doesn't identify that. It doesn't identify a foreign antigen if there's not one. Right? So you want to, okay, here's what it's on the test. You want to focus. The mom is negative. The problem is the second positive baby. Y'all got the story? The mom is negative. It's the second positive baby that's a problem. Okay, can we move on? The next part of the notes talks about abnormal immune system functions. Okay, so the next part is when your immune system, the word autoimmune is where your immune system infects or attacks you. Your immune system attacks you. So it doesn't recognize that you're autoimmune is where your immune system attacks yourself. Okay, so the first one, the first thing is allergies that we're talking about. Allergies is where something like a piece of pollen gets into your body, and your white blood cells identify that as a pathogen. Well, it's just a piece of pollen. It's not a pathogen. But your white blood cells attack it, and right in your nasal cavities, you have mast cells, which release histamine. What is histamine? Inflammation, right? Y'all remember seeing that on part 1? Histamine is inflammation, swelling. So you have these cavities in your skull, and when the pollen comes in, the swelling occurs in those cavities. That's why you get like a sinus infection and then runny nose. And it's really, your immune system is overworking. It doesn't need to destroy that pollen. Your immune system is working way too hard over just a piece of pollen. So that's what allergies are. It says a false alarm. Okay, so now we're talking about autoimmune disorders, and that's where your body attacks you. And the first one is lupus. And lupus is where your immune system infects your kidneys. And you can see on this picture, it's characterized by the butterfly rash. I actually know a girl that has lupus. I went to college with her. And she, like her immune system would attack the organs. But and I don't know if this is normal or just her, but they actually put her on chemotherapy. And even though she didn't have cancer, chemotherapy kills quickly dividing cells, right? So the chemotherapy killed her immune system. And I think she went through 2 rounds of chemotherapy. She was like a softball player. She was like, had good health. Well, except when she was on the chemotherapy, she didn't. But she went through 2 rounds of chemotherapy, and she didn't suffer from lupus anymore. Like, she may have still had it. I don't think they cured it, but it didn't act up anymore. And they can take medicine with lupus, and it's not a big deal. Do y'all know someone with lupus? It's pretty common. Okay, and the rash is not always there. The rash goes away. Okay, the next one, it talks about, arthritis is next, rheumatoid arthritis. That's where your immune system attacks the joints of your fingers. So arthritis is an autoimmune disorder where your white blood cells attack and break down the tissues in your fingers, and it hurts. The next one is diabetes type 1. That's where your immune system attacks the pancreas cells that make insulin. So you don't make any insulin, so you have to take insulin. Type 1 is like child's diabetes. You get diagnosed under the age of 20. Yes, ma'am.
So, like, anything that you're born with is basically autoimmune disorder?
No, it's got to deal with white blood cells. Good question. Okay, and then MS, you've seen this picture before. MS is where your white blood cells attack the myelin sheath. Right? The white blood cells attack the myelin sheath, and so when the nerve impulse travels down the axon, it doesn't travel as well, and it like burns the muscles. Okay, the next part of the notes is immunodeficiency, where like you were born or now your immune system doesn't work. So autoimmune was where your immune system worked. It just beat up the wrong things, like your joints. Now we're talking about where the immune system is inefficient. The first picture here is a kid with SIDS, and that's someone who's born with no immune system. Have y'all ever seen that movie? It's like The Boy Who Lived in the Bubble or Bubble Boy or something like that. I've never seen it either, but I've heard kids talk about it.
What you talking about when he lives?
Is it a Lifetime movie?
When he lives in the little thing?
Yeah, he lives
And his mom keeps him in the house, and then he escapes, and his bubble goes around the room.
Yeah, he lives in a bubble, and his mom like wears a mask and hands him his food, and then goes out real fast. Can't ever go anywhere because he doesn't have any immune system.
But it finds out it wasn't true.
Do what?
It wasn't true at the end.
Oh, wow, that's horrible. Okay, Hodgkin's lymphoma is cancer. I don't have a picture of that. Hodgkin's lymphoma is cancer in the lymph nodes. Your lymph nodes are on your armpits and throat. We talked a little bit about those. They are part of the immune system, right? So they might get swollen when you have an infection. Well, the Hodgkin's lymphoma is where the lymph nodes are destroyed in the cancer. Have you ever heard of non-Hodgkin's lymphoma? Maybe somebody who has cancer, you've heard them say that. Well, Hodgkin's lymphoma is where the lymph nodes are destroyed. Okay, stress. I don't have a picture of anything like that either. Stress is when you're worried about something, like an AP bio test, but worse. Something that you worry about for a long time, for like months. I know you're worried about my test. Okay, maybe not. But stress, like if you maybe are in college and having a really, really rough semester, like a long time stress, you may get the flu, and then pneumonia, and then a urinary tract infection. And what it is you're spending all your energy worrying about what it is that you're stressed about, and not enough energy on making new cells. And like, you know how when you're stressed out, you don't sleep good? Well, you've got to sleep good for the immune system to work right. You've got to eat good for the immune system to work right. And when you're stressed out, I feel like you don't eat as good, you don't sleep as good, you're spending all your energy worrying and not enough energy working on your immune system. Stress will kill you if you let it. Okay, AIDS, I do have a picture of HIV. This is the HIV virus. So remember we talked about the difference in HIV and the difference in AIDS. And someone with HIV or AIDS, their helper T cells are infected. Do y'all remember that? Helper T cells are infected?
Yes.
So they don't have, remember the helper T cells are infected, so they don't release IL-2, so the T cells and B cells don't work. Okay, well, AIDS is a virus, and it has RNA inside it. RNA is inside the virus. Well, your body has DNA in it, right? It makes RNA, but our body makes DNA, RNA, protein. DNA, RNA, protein, right? Okay, so when the HIV virus incorporates its RNA. It also incorporates an enzyme that makes it go backwards. So it goes from RNA to DNA. And then it goes DNA, RNA, protein. DNA, RNA, protein. So the HIV vaccine takes over the DNA of the helper T cell. So it incorporates its RNA. It makes DNA with the enzyme that makes it go backwards. The name of that enzyme is the reverse transcriptase. And that makes perfect sense because transcription is DNA to RNA, right? Y'all remember that? So reverse, so it makes DNA. So the RNA makes DNA. And then the DNA, RNA, protein. DNA, RNA, protein.
What's bad about that?
Nothing. It's just the virus is just bad because it takes over the helper T cell. We just classified it as a retrovirus. So the word retrovirus is a virus with RNA instead of DNA. That's all. I was just telling you the classification of it. Okay, so highlight retrovirus on AIDS, and you want to know that it infects helper T cells. Okay, so the next part is all about plants. Plants sometimes have gene-to-gene recognition. All that means is where the plant can identify the gene of the pathogen, and it becomes resistant to it. That's all that means. Okay, elictors. Elictors are a message, a chemical message sent in plants. Plants, do y'all remember what xylem and phloem are in plants? Yes, phloem is where the sugar is transported down. A little by the way. Okay, so elictors are transported in plants. All it is a chemical message saying that there's a part of the tree that's infected. So they don't have a bloodstream, but they can transport in their phloem. Elictors, chemical message that says that there's a sickness, and they release oligosaccharides, which is like an antibody. Oligosaccharides are like antibodies. Right there on that elictor part. You see it says elictors. These plant cell wall pieces, they're plant cell wall pieces indicating that there's damage. So it's like a chemical message saying that there's a pathogen. Okay, and then oligosaccharides are released to cells, causing them to release the antibody. Phytoalexin. Phyto means plant. Okay, these next two are important. You want to know these next two things. Hypersensitive response. Okay, when you get an infected leg, let's say you got a big infection on your foot, where's that infection going to go? To the rest of your leg and then up your body to your other extremities, right? It's going to go all over. And if you got a really, really, really bad infection in your foot, then your foot would turn black. That would be the skin dying. That limb is going to die. So you would have to go to the doctor. Hopefully you'd already be at the doctor. And you might have to get it amputated. And in order to save the rest of your body, you cut off the leg. Yes? Y'all following me on that? Did anybody watch Grey's Anatomy last week?
That's what I was just saying.
When that snowboarders, both of his legs were black, and they cut him off, and then of course, because he was a professional snowboarder, his life was over.
Yeah.
But the doctor was trying to explain to him, she saved his life by cutting off his legs. Okay, plants can do that naturally without the doctor. They can, if there's a part of the limb that's infected, they can break off that limb, and that's called hypersensitive response. Yeah, we talked about this a little bit. Plants can amputate their infected part naturally, and that is so cool. It says massive release of phytoalexins, that's the antibodies, causing a sealing off affected leaves. It says trying to keep the pathogen from advancing further. This creates a dead zone that the pathogen may not be able to move past. So can y'all see how it's kind of doing it in this picture right here? The hypersensitive response. It's like that part of the leaf is gonna break off. It's killing that part of the leaf so that the pathogen can't move forward.
Does it grow back?
Yeah, it grows back for sure. But now in ours it doesn't. If you get your limb, it's not. Then if you get yours, the cells are going to grow until they touch again. So they're not going to grow like a new finger. Okay. And then the last one, systemic acquired resistance. That's where the plant is resistant and it releases salicylic acid. Okay, so the whole plant becomes resistant to the pathogen. Questions?
Can you go back over A?
Mm-hmm.